our new speaker. I forgot my notes, but starting with important things like the restrooms are right outside the front door. If you want to receive email notification of upcoming programs, please sign the email lists, either for the library or Alliance for Science. We are so happy to partner with Alliance for Science, Hawaii Alliance for Science, to put on science programs for the community. And this is our third program. We're lucky to catch Dr. Freeman, who is visiting his mother, who lives in Pu'u, Mrs. Freeman, his wife and young son with the fish shirt are in the children's department at the library. So we welcome you, and I'd like to introduce Sarah from the Alliance for Science to introduce herself.
Hi, thank you all for coming. Thank you, Carolyn, and the library for hosting. My name is Sarah Thompson. I'm with the Hawaii Alliance for Science. This is a partnership between the library and the Hawaii Alliance for Science to bring these programs here. I want to welcome Chris Freeman. He is speaking tonight for us, and that all happened because the Hawaii Alliance for Science started to organize the March for Science, which happened in April of this year and both last year as well. And was part of our organization committee for the March for Science, and so that's how we got to know her. And we told her about this Science at Your Library program, and she offered up her son as a speaker, so we're happy to bring him here. I want to recognize also Jan, who is part of the Hawaii Alliance for Science as well. And there may be some other people that show up a little later tonight. Feel free to learn more about us by visiting our website, HawaiiAllianceForScience.com. There's more information about the organizers as well as the next speakers that are coming and our previous speakers. We have videos of their performances, speeches as well. And thank you guys all for coming. And I would give you a history of Chris. I really don't know that much about him, so I'm excited to learn more about his work tonight. So everybody, welcome to Chris Freeman, Dr. Chris Freeman.
All right, well, thank you all for coming. So just as a little introduction, so I currently live in Charleston, South Carolina. And the work that I'm going to talk about tonight, I was a postdoc down in Florida at the Smithsonian Marine Station, which is sort of a research offshoot lab of the Natural History Museum down in Florida. And so right now I'm an adjunct professor at the College of Charleston, but I still do a lot of research in the Caribbean. And so today I'm going to talk about sort of going to give you an introduction to a smaller group of organisms than you might know in the ocean called suspension feeders. And so sorry. You did?
You're back, you did.
Yeah. Wait, it's too far. Which button is it?
electronics are not working very well.
Is it gonna work?
It's my mistake.
Yeah, so I'll just click on here. Let me test it out.
I think.
Okay.
You have the library back there.
Sure. So this might be a really weird talk. I'm gonna stand in the back of the room, but it'll work.
You can see how your slides are
Yeah.
in the back.
Thank you. Let me touch it. Okay. Sorry about that. Let's see what happens. Okay. So yeah, I'm gonna talk about suspension feeders. Hopefully, let's see. Okay, so if I were to ask you to think about predators or grazers, or think about predation or grazing, you might think about a couple things. You might think about gray white sharks catching seals in midair, or you might think about cheetahs chasing prey in Africa, or even cute little cows munching on grass. And so what I want to introduce you to are another really important grazer community, which are suspension feeders. And so these organisms here, these include several different types of organisms. So you have the bottom left, you have a sponge, which is what I've mostly studied. You also have tube worms, rhizomes, tunicates, bivalves, all sorts of different organisms. And so these organisms have really interesting adaptations that allow them to filter out or catch particles from the water column, and they consume those for food. And so I'm going to talk to you a little bit about who these organisms are and the role that they play in some of these marine systems. And the first question is, what do they eat? And so they eat really tiny stuff. So this is a beautiful micrograph of some diatoms that have been colored. So we're talking about organisms that are about the size of diatoms, so quite small. And this is an image of all different types of small organisms, different planktonic organisms that live in the water column. And so this is a huge range of critters. So in the center there, you have really tiny organisms, and some of those include organisms that are almost the size of bacteria, so very tiny. There's also some diatoms in there, and as you move out from the center, you get into zooplankton, which are a little bit bigger. And so these suspension-feeding organisms feed in a pretty big range, anywhere from about 100 to less than 1 micron. And so a bacterial cell is about less than 1 micron, so very tiny. And 1 micron, just to give you a frame of reference, is 0.0004 inches. So if you think of your hair size, so one strand of your hair is somewhere between 50 to 80 microns in size. So very small. And these suspension feeders are very efficient at removing these organs from the water. And so the other question is, how do they eat? How can they possibly remove these tiny little critters? And so, like I said, I've mostly studied sponges. And so I'll give most of my examples through them. And so this is a cross-section of a couple different types of sponges and how it's sort of a rough representation of how they bring water into their bodies. And so I can't really point out, but if you look at these arrows that are coming into the sponge, so the sponge is this sort of vase-shaped thing. The arrows are coming in, and then at different stages, especially the one on the bottom right here, you look at those little sort of chambers inside. Those are, you can think of those as like filtration chambers. And so the water's coming in, all these cells are doing their thing, they're filtering out the bacteria from the water, and they're allowing the sponge to eat that up. And so it's one thing to talk about it, but this is actually an image of what that would look like. So this is a sponge from Panama, and it's a pretty small sponge. Those brittle stars that are attached to it are maybe a couple inches in size. And this sponge is not very big, it's an encrusting sponge. And so this yellow dye is called fluorescein dye, and you use it to visualize water movement. And so I'm going to insert a little bit of this dye on the outside of the sponge, and the sponge is very quickly going to take the dye, and you're going to see it shoot out of one of those little holes there. And so the water that's coming out of that hole has been filtered of at least something, bacteria, phytoplankton, something like that. So you'll see how quickly it can happen. So within a couple seconds, the sponge is taking up that dye, it also has water in it, and it's filtered it out, and it's come out of these larger holes. And so that's how quickly sponges are filtering water. They're doing this all day, every day. So they're very, very efficient at taking these bacteria from the water. Let's see if I can go back to. Okay, so they can trap, this varies across species of sponges, but as a general rule, sponges can trap a ton of bacteria in the water that they filter, and they can pump about 10,000 times their own size, volumetric size, in water in 1 day.
Is that a colony or a single organism, the sponge?
It's a single organism.
Mm-hmm.
Yeah.
Mm-hmm.
And so, if you think of a residential swimming pool, it's about 10,000 gallons of water. So a sponge about the size of a gallon of milk can filter in 1 day a residential swimming pool. Now, you guys have sponges in Hawaii. The world's largest sea sponge was actually found up in the northwestern Hawaiian Islands a couple years ago. And so they said that this is the biggest one. It's about the size of a small bus, and it could be thousands of years old based on the fact that it was found in the deep sea, and it likely grows very, very slowly. And you also have sponges in Hawaii that are in more shallow water, but they're certainly not as abundant as what we see in the Caribbean. Now, sponges are prolific across the globe, so you have them pretty much in every ocean, the Great Barrier Reef, they're in the South Pacific, Antarctica even, and also the deep sea, so they're very widespread throughout the globe. But I'm going to focus mostly on this area in the Caribbean, so all the way down from Panama, all the way up to the Florida Keys. That's where I've done most of my work, so I'll spend most of this talk on that area. I'm going to skip this video because you guys know why. But this is what the Caribbean reefs in some areas look like. So this was a reef off the coast of Honduras that was a really pristine reef, far from any development. And a lot of these really colorful things that you see in this image are sponges. So the red things, most of the orange ones are corals, but the purple ones and some of the blues, they're really intricate, rope-shaped, sort of massive sponges. And so they're quite abundant. Here's another image from the same reef here. And so in the Caribbean, you have roughly 500 species of sponges in the basin. And to give you an idea of how that compares globally, there's about 8,800 sponge species on a global scale. And in the Caribbean, about 16% of the bottom on average is covered by sponges, but there's a pretty big range across sites, so anywhere from 2 to 75% of the bottom is covered in sponges. They can be really big. So this is a giant barrel sponge. Zesta sponge Vita. And this person next to it is probably 5.5 feet tall. So this sponge could be hundreds of years old, and you can imagine how much water is filtering every day, and the role that it's playing in this system as a filter feeder. And one of the questions that we've been wondering is, why are sponges so successful? Obviously, this translates to a global scale, but we've been focused mostly on the Caribbean. And so one of the ideas is that it's because of microbial symbionts that live inside of them. And so we first focused on cyanobacterial symbionts. So this is what a cross-section of a sponge that has a lot of cyanobacterial symbionts looks like. And so the black thing on the right is a giant canal, so that's where water's going to come through for filtration. The greens and the yellows, those are sponge cells, a part of the skeleton. But then all those little orange dots, those are all cyanobacterial cells. And so if you zoom in on one of them, they look like this under an electron microscope. And so these cells, these cyanobacterial cells, have the machinery to perform photosynthesis. So they're really good at fixing carbon, and they can actually pass on some of this energy they get from fixing carbon to the host, and potentially benefit the host. And that was our first question. And so we started out by just looking at how prevalent these cyanobacterial symbionts were across sponge species in the Caribbean. And so we surveyed 60 species. And so on the left side over here, that axis is chlorophyll A, which is just you can think of it as the abundance of these cyanobacterial symbionts within a sponge. And so about 20 of the species had what we call high levels of these symbionts, and the rest had pretty low levels. And so there was quite a bit of variation across species in the Caribbean. And that's interesting, but we were really wondering, do these symbionts actually benefit their host sponges? And so my PhD advisor loved to do these shading experiments. That's sort of like a rite of passage in the lab. Your first summer you had to do one. And so he sent us to Panama, and we set up these shading experiments where they're really easy. You take a sponge, you split it in half. One half goes under a shade canopy, which is in your upper left. The other half goes under an open canopy, which is your bottom right. And so the shade canopy blocks 93% of the light. And so if you're a cyanobacterial symbiont, you can photosynthesize less, and so theoretically you're passing less energy on to your host under the shade than you would in the open canopy. And so from this shade experiment, we measured cyanobacterial abundance as our first metric. So the y-axis here on the left is chlorophyll A concentration. So once again, the abundance of cyanobacterial symbionts. And so you have initial in gray, control in open, and shade will be shaded. So for the first sponge, you see that initial and control symbiont abundance was identical, but in the shade it was greatly reduced by the black bar there. Same thing for this species, and same thing for this species. So all three of these species had really high levels of cyanobacterial symbionts to begin with, and they were all decreased in the shade treatment. This last guy here we added sort of for fun, because we knew this species didn't really host these symbionts, so it has roughly 40 times lower concentration of these symbionts in its tissue. So we kind of added it as a control. And so how did this affect the host growth rates? It affected their growth rates a lot. And so your y-axis is relative change in wet weight. And so for these three species that had really high levels of cyanobacterial symbionts, their growth rate was decreased drastically, so that you can see that the shaded bar is much lower than the open bar. And this species here that did not host cyanobacterial symbionts actually grew more in the shade. So you can see that shade didn't impact it at all.
Why would it grow more?
So we think that what happened is it just, we moved it from a new site. We moved it from an old site to a new site where the experiment took place. So there might have just been more food at that site for that particular sponge, since it tends to filter feed more. And so it's looking like there's some variation in the benefit across sponge species, but we wanted to look at this more, and so we did a tracer experiment. And I'll walk you through this, let me know if you have any questions. But if you take your cyanobacterial symbiont and you feed it a source of carbon that it can fix during photosynthesis, so you use the sun's energy to take up this is sodium bicarbonate, so it's baking soda, which everybody has in your house, but that 13C means that it has a particular label in it that we can measure later on. And so we feed it this carbon source, it's exposed to the sun, it fixes it, and then we can measure, using a very fancy instrument, we can measure the concentration of this 13C in the cyanobacterial tissue, with the expectation that it's going to increase a lot because it's going to be fixing this carbon. And then over time, we're hoping that there's going to be a transfer of this photosynthetically fixed carbon from the cyanobacterial symbiont to the host, and that the host tissue is also going to become highly enriched. And so this study allows us to look at the efficiency of these symbionts and how good they are at transferring energy to their host, how much of a benefit they're giving their host. And so you think of it as a race. So we took 6 species of sponges, we exposed them to this 13C labeled bicarbonate, and we let them run. And then at the end of the experiment, we measured their 13C levels. You had a winner, which was the sponge that gave its greatest benefit in the form of 13C to its host. So we measured 13C in the host tissue. And we had, I wouldn't say the loser, but it's the slower sponge, so the slower symbionts, that had the lowest levels of 13C in its tissue and gave its host the least benefit. So what this looks like if you actually look at the data, the y-axis over on the right side is the concentration of 13C And then the x-axis rather is a measurement of productivity, which you don't really need to know too much about, but know that there's a cutoff there of 1.0. So anything above 1.0 means that those cyanobacteria are really productive. They're really giving a good benefit to the host. And so this is our friend here that doesn't host cyanobacterial symbionts. So you would expect a really low benefit to the host. And indeed, you can see it had almost zero 13C in its tissues. So it's giving a very low benefit to its host sponge. And then you have these other species. These are all species that host fairly abundant communities of these symbionts, but there's tons of variation in the levels of 13C in their tissue. And I really just want to point out this one here was incredibly productive. So it was transferring a ton of this 13C that it had fixed photosynthetically to its host, so a really big benefit. And it was also very, very productive, so much higher than 1.0. And this just means that some symbionts are more efficient than others in this pathway, so this carbon pathway here. And what we found was that what really mattered here was the actual identity of the symbionts, of the cyanobacterial symbionts that were in each of these sponges. So it varied a lot across these different species. Think of it as sort of like a different strain of these symbionts in each of these different hosts. And so that particular strain really was what was driving these trends in this carbon transfer. And this was important because about the same time, so this is my representation of a Caribbean reef, beautiful Caribbean reef, just completely full of sponges, right? And so about the same time, people started looking at the whole microbial communities that were present in these sponges, not just the cyanobacterial symbionts. And what they found was they were using more advanced techniques to profile these communities and see who's really there, what their relative abundances were. And so they found some species had incredibly high levels of symbionts, both in abundance and also just the different types of symbionts. So the different types of symbionts here is defined as richness. So they call these HMA, or high microbial abundance. And then your other group was called low microbial abundance, and these were all thought to be, they had really low abundance of symbionts, and also maybe like 2 to 3 different types of symbionts. So not really too much exciting going on in the LMAs. They thought maybe that it was just actually a lot of ambient bacteria from the seawater that were present in their tissue. But then more recently, even more advanced methods have been used, and they found that these LMA, there are actually HMA species that have really low levels of different kinds of symbionts. So it looks like they have a very specific group of symbionts inside of them. And then there were actually some LMA species that, even though it's a sponge, and so it's sitting in the water, filtering water all day long, it really only has a single symbiont type within its body. And so this suggests that the sponge is somehow controlling who its true symbionts are. And so it sort of set the stage for the idea that LMA species, there's a lot of selection for specific symbionts in these groups as well. So the symbionts in LMAs are likely doing something really important. And so it didn't end there. This study it's actually a colleague of mine, Eason and Thacker, at the bottom, they surveyed 20 species, actually these 20 species of Caribbean sponges, and they found that each of these sponges, even really closely related ones, had vastly different microbial communities in them. And so they found that 70% of the dissimilarity in the microbial communities across these hosts was driven by host identity. So that might be a little hard to sort of fathom, but what it means is that even species that are in the same genus had vastly different communities. And so there's something that's structuring this selection process for these different communities across these species. And one idea is that having unique microbial communities allows these species to diverge in the resources they use. So if you think of these sponges here, and if you imagine these 20 species of sponges living on a single coral reef, maybe 20 feet by 20 feet, which isn't that far-fetched in some areas in the Caribbean. And so they have access to all of these nutrient sources that are in the water column. So you have all the things they could filter out. They also have a lot of sources of inorganic carbon and nitrogen that they could possibly use with the help of symbionts. And so if these sponges have unique microbial communities that allow them to access different portions of that nutrient pool that's in the water around them, then this could really benefit them and allow them to all coexist in one area. And so I sort of define this as this, just unique resource use due to their associations with microbial symbionts. And so in the next few slides, I'm going to address this by talking about the microbial symbiont communities that we found in some of these sponges, and also their resource use.
I don't know if you're going to get to this, but are they born with the symbionts, or do they acquire them out of the water column?
So it's a great question. People are looking into it, so there is evidence of vertical transmission where the symbionts are actually transferred from the parent sponges to their offspring and gametes. But for the most part, it's sort of a mix. It's called like a leaky transmission. So there's a mixture of both horizontal transmission, where they're getting it from the water column, and also vertical transmission, where they get it from their parents. So we started this at a really special place called Bocas del Toro, Panama, where there's another Smithsonian station. And so we went to this one site called Saigon Bay, which is right next to the research station, and it's just completely full of sponges. And so we sampled these 15 species. I just put this up there to show you, but the red sponges are HMAs, previously thought to be high microbial abundance, and the blues are LMAs. And we measured the isotope values of these sponge species. I'll tell you why in a second. So stable isotopes, a brief little introduction to them. They're just atoms whose nuclei differ in the number of neutrons. So you see carbon-13 has one extra neutron. It doesn't make that big of a deal, but you can call carbon-12 the light isotope. You can call carbon-13 the heavy isotope, because it has one extra neutron. So that neutron weighs almost nothing, but in processes like photosynthesis, that extra neutron actually weighs it down and causes it to have an influence in the isotopic label that's transmitted from inorganic carbon to the cyanobacteria. And so what we use this for is we want to know where the atoms within an organism come from. So if you think of yourself, we can measure your stable isotope ratios, and we can gain a sort of an understanding of where you're getting your carbon and nitrogen from. And I have a cool figure next that'll talk about that. And so your carbon isotope values tell you about the carbon at the base of the food web. So where are you getting your carbon? Nitrogen tell you about your nitrogen sources, and they also allow you to estimate where you sit on a food web or trophic position. And so think of this as a time-integrated estimate of diet. So you're accumulating your isotopic signal on a day-to-day basis from all the foods that you're eating. And so I like to show it this way. This is how an isotopic diagram is shown. You have your delta 15N values on your y-axis on the left, and your delta 13C values down on the bottom. And so if I were to take a sample from most people in this room, it could be hair, nails, whatever. It's a very small sample. And I analyze the isotope ratios, you might get a figure that looks like this. And so you could break this down to different groups. So the bottom here would be vegetarians that feed themselves at the base of their food web. And then you move up to more omnivores, carnivores, and people that really focus on higher trophic level fish. And so for the delta 15N values, it increases by a set amount with every trophic level. And so in your carbon for humans, it's really going to be determined by how much either rice or wheat or corn you consume. So if you're a vegetarian that eats mostly corn, you're going to be on the right side of this figure at the very bottom. And so this division of the people in this room, if we were to measure isotope values, is a good way to look at how different our diets are. And so we're using a similar idea to this with the sponges to look at how different their resource use is. So for sponges, you also have nitrogen and carbon sources. You just have a lot more potential sources because they have the symbionts that can help them utilize different sources. And so what we're hoping to find is, so this is sort of what people propose with this. If you just divide sponges into HMAs or LMAs, you might see something like this. But what we're hoping to see is something more like this, where sponges are in different portions of this figure. And so that would suggest that they have different metabolic, physiological, etc. adaptations that allow them to utilize and also process resources differently than their coexisting sponges. And so this is, I think it's 75 points from Saigon Bay from all the sponges we collected there. So you can see there's some variation. If you color code these by species, I like to use this instead of averages. I can talk about it at the end later because we did some additional analyses too. But you see that the same colored dots tend to be pretty closely linked to each other. So there's not a lot of variation in these isotope values within a species. Most of the variation is across the species. And in fact, 85% of the variation in the placement of individual dots in this figure is driven by a species. So what that means is that, just like I showed you with the human graph, sponge species in general are present within unique sort of space within this graph that suggests that they're utilizing and processing unique resources of carbon and nitrogen at this one reef. And so that's one site. We did this at a couple more sites in Bocas del Toro. We did it in Honduras, Belize, and also the Florida Keys. So it's 25 sites in total. And at all these sites, we found similar trends. So these are regional averages. And then the Caribbean average was about 86%. So 86% of the variation in the placement of these individual samples within that carbon-nitrogen graph is driven by host identity. So there's lots of individual species are widely dispersed within this figure, which suggests that they're utilizing and processing unique sources of carbon and nitrogen. And so what about the microbial communities? Well, we threw, it's actually it's a lot more complex than just throwing, but we analyzed the same samples using this very fancy instrument, which is an aluminum iSeq. And so this tells us, this. And so this figure is a little complicated, but these are, actually I don't want to touch the thing, but so your x-axis on the bottom, those are all sponge species. So that's 20 different sponge species. And your axis on the left is the relative abundance. And so each of these colored dots on the right side are all different bacterial groups. And so what this instrument allows you to do is it allows you to take a sponge sample and it tells you what bacterial groups are in it and what the relative abundance is. And so I can't point at it, but if you look at sort of the middle, so if you count 1, 2, 3, 4, 5, 6, 7, 8 from the right, you see that one species that is almost all green right there, Itracoda birotulata. So that's an LMA species that's actually dominated by, it's like 98% one bacterial group. So you can see how that looks. There's just pretty much that one green bar there, which just means that that species is dominated by a single bacterial group. And if you move over a little bit further to the left and you go, actually we can just use the farthest one on the left, Ilacroix crassa, if you can read that. You see that there's several different colors there, probably 10 to 15 different colors there. And so that's an HMA species that does indeed have many different types of symbionts. And so that's what this really cool instrument tells us. It tells us what's there and what relative abundance. Yep.
Are there any bacteria species that are more likely to be the only bacteria present?
That's a great question. So these, I think to really answer that, you'd have to get, so these bacterial groups that I was talking about, these are like bacterial phyla, so they're really broad groups. So if you wanted to really look at that, I think you'd have to break it down into more specific types of bacteria. But there are ways to do that now, and so people are looking into that kind of stuff more. That's a great question though. Sort of looking at the idea of like who are the true, so if you were to do like who are the true symbionts and what are very specific actual bacterial species or bacterial strains doing for the host. Yeah.
Have you guys separated each of these individual symbionts and tested and found any reason why one would be dominant over any other?
We have not, no. I don't know if, I'm trying to think if anyone is currently looking at that. One thing that's tough is that these symbionts don't do well in culture generally because they're really relying on their host, and so it's difficult to separate them and keep them alive for experiments like that, but there are some ways to do in situ experiments where you can start to look at that.
Can the host lose or gain these bacteria?
There's some dynamics of the communities over time, but
Say with temperature change or changing in the environment, would that stress them to do that?
yeah. So sponges are generally thought to be. There's some data to suggest that they're more robust than corals, so they're not. Not all sponges bleach or anything like that. There are some diseases that have caused shifts in the microbial community compositions, and that could be caused by a few different things like exposure to some pathogen or pollution, different things like that.
Well, would it be good indicators for the health of the reef or vitality in that
Just
respect?
sponges in general.
Yes.
Yeah.
And what would be the indicator, say, the bacteria
Of the bacteria, I think if you had, would probably be just a better indicator would be how the abundance of sponges or the presence of specific species of sponges shifts over different sites or over time. So you could use sort of their surveys of them to correlate with shifting environmental conditions, see if there was a relationship. And so we analyzed all of these samples that I talked about before using this instrument. We found pretty much the same thing with microbial community composition, so really host specific. Even species in the same genus had vastly and significantly different microbial communities. And the really big question here was if there is a link between the composition of these microbial communities and the isotope values, so their sort of metabolism. And so we looked for a link between these trends. So is there a link between isotope value trends and the community composition trends for the microbial symbionts? It turns out that there was for nitrogen, which is really interesting because a lot of the work that's been done with sponges in the Caribbean to date has focused on carbon predominantly. But it makes sense that there is this relationship with nitrogen because on coral reefs, nitrogen is generally thought to be a limiting nutrient. And so there could be this, that could be driving some of this divergence in microbial community composition and function. And so in conclusion for this part of the study, of these 20 species that we looked at, we did see some evidence of what we're calling functional divergence, so unique trends in their isotope values. And this is really the first step in this. I wanted to talk about something else in addition to sponges for this, so I kind of cut this part a little short, but I do have some other data that I'd be happy to talk to people about after this that supports this even more. And we actually found out that my colleagues and I just got a National Science Foundation grant to look at this more. This is really the very first step in this, and so we're going to expand it quite a bit. And mostly that's because of this potential link between metabolic divergence and so the use of unique resources and microbial symbiotic communities in this ocean basin. And so, let's see, I have one more thing. So couple of things that we're looking at is the isotope values do suggest that some of these species can, because they have these symbionts, they can fix nitrogen, which would be a unique source of nitrogen instead of just getting it from filter feeding. Another would be recycling nitrogen. So the host metabolism is, of course, releasing nitrogen into the sponge. And so some of these symbionts can take that up and allow the recycling of it instead of just the loss of it. And a big topic now in sponges is the ability of species to take up and utilize dissolved organic nitrogen or carbon. There's something called the sponge loop, which is really big in sponges right now. And it's actually got published in Science several years ago. And so it's the idea that sponges can take up dissolved organic matter and ultimately they release all of that nutrients that they've gained from the dissolved organic matter and release it as particulate matter, and then other organisms around that can take that up and survive. So there's this loop between dissolved and particulate that feeds these communities and keeps reefs in general healthy and very diverse. So I would be happy to talk more about the sponge stuff afterwards if anybody would like to, but I want to jump up to some subtropical stuff. The Indian River Lagoon. So I did my postdoc here at the Smithsonian Marine Station. So all of the research that I'm talking about today was done here. And I want to just introduce you to this. I got most of my photos from that first slide from this system. So it's thought to be one of the most diverse estuaries in North America. And it's really quite beautiful. So you have, once again, tunicates, bivalves, rhizomes, tube worms, barnacles. Right in the center, in the very top, those are really super little tiny bivalves. Well, one's a bivalve, one's a gastropod. And so these are all epifauna, or fouling organisms that are suspension feeders. So these are things, if you have a boat, you hate these things, right? Because they're all over the bottom of your boat, they're slowing it down, you have to get scraped off. But they're also really important. And so in the IRL surveys, the IRL is the Indian River Lagoon again. So surveys at about, this is 30 sites. Species richness is just the number of species. You can see that it varies across these sites, but it's generally really high. Like B3 has about 35 species in a single site. And so in total, surveys across the lagoon found over 150 different species of these fouling organisms. So it's really quite diverse.
Is this freshwater or marine?
It's a mix, it's brackish. So it has some, actually don't have an image of the system itself, but it has, it's fed by a lot of different rivers. Then it has some inlets to let marine water in. And so it's, there's a little bit of salinity gradient, but it's not too bad. It's a beautiful system, but it has its problems. So this is a brown tide that hit the system a couple years ago, and they've been coming back repeatedly. And that causes fish kills. In this case, it was a massive fish kill up in Cocoa Beach. So thousands and thousands of fish dead. There's also some algal blooms that are harmful. So microcystis blooms that can be really damaging to humans, to human health. And there's, it's highly debated what's causing these blooms. I don't know if you guys are familiar with Florida in general, but Lake Okeechobee, they have to empty it out every now and then when they have a lot of rain. And so all the nutrients and issues from that freshwater system are dumped into inlets, which then feed into the lagoon. And that's thought to cause a lot of these algal blooms because of all those excess nutrients that are being released. So there was a really big bloom in 2011, and you can follow it through this mosaic image here. So these, the red areas are all areas of really high algal concentration. So really, you think of red as really bad in this case. And just to give you sort of an orientation, this is massive, right? So this is about 215 kilometers long. So it's a really big system. It's not small. And so this super bloom lasted about 8 months, and it was dominated by these two species here, Synechoca calis and chlorella. You don't need to remember those. I'll keep reminding you. But this bloom did everything you would expect it to do. It killed fish, killed 60% of the seagrass in this northern part of the Indian River Lagoon. Manatees were dying. It was a big mess. And
How's
so,
that map? How did you map that?
so this is based on satellite images.
Okay.
Yeah. So the author over here, Kamarowski, they just used satellite data to figure it out. And so in the IRL, everybody loves bivalves because historically they were really the dominant and most important filter feeder or suspension feeder. And also they have a huge industry for these in this system. And so we wanted to start by looking at whether or not, even though these particular 4 species are really not present in large numbers like they used to be, we wanted to see if they were there, would they be eating these cells? So would they, if we brought back the bivalves, could it help prevent an algal bloom? You can think of it that way. And so we actually were able to culture algal cells from the super bloom that we had obtained from a colleague. And that looks like this. So we have chlorella and Synechoca calis in the lab in these pretty big beakers. And so we were able to culture these up to these really high concentrations that we could use in experiments. And then we fed these to these bivalves of interest in the lab. And did it work? It did. So I'm kind of cutting that part short because I'm going to focus on something else. But the problem with this experiment is that it's definitely interesting that if we could bring these back, they would be capable of eating these bloom species, but they're not there anymore for a reason. A lot of that's because the system, the environment there just doesn't support bivalves. It's too silty. Their gills get clogged up. They just can't survive like they used to there. And so if they were there, they might be able to do something, but a lot of work's been going into oyster reef restoration in this system, and quite a few of these reefs end up failing in the end because the system's just not good for them anymore. And so what we see in this system are these beautiful, complex, diverse, really beautiful communities of suspension feeders. And so instead of focusing on bivalves, we were wondering, what are these guys doing? They've got to be doing something, and so let's test this. So we put out these settlement panels. These are about 2.5 by 2.5 inches, so not very big. And it's just flat PVC that have been sort of sanded to give it some structure. We put them out at 15 sites throughout the Indian River Lagoon. And then 5 months later, we came back to get them. And this is what they look like. And so these are all organisms that have settled and started to grow on these panels. And you see, really what I want you to notice is there's tons of variation in the diversity, the colors, the percent cover, the number of species, all the keywords that you want to see. And so you have, like in the upper right corner there, those are mostly barnacles with a couple of tube worms. You might have like 2 or 3 species up there. The panel next to that with those big black blobs, that panel had 35 species on it. This is a panel that's, this 2.5 by 2.5 inches with 35 species on it. And so this was a perfect way for us to look at the idea of, is community composition or diversity driving trends in the potential removal of these bloom algae. And so we built this really cool manifold system in the lab, in the wet lab. So each of these panels had its own little home, its own little bucket, and these were fed by separate water systems, and they had a little aerator. And so we did that. You can zoom in here to see it. So the panels were held within these. They were very happy. Nothing died over the course of the week, and it seemed to work out really well. The experiment was really simple. We used these cultured species, the chlorella and the Synechococcales. Here's your little panel, my favorite with the 3 black blobs. And these, once again, these were the super bloom cells. We fed them to these panels. Really like not bloom concentrations. We didn't want to kill everything off or overwhelm it. So we fed them at lower concentrations of 3 times 10 to the 4. And then we took samples. So we immediately took a sample, a 1 milliliter sample at 0. That's your initial. And then we took samples over the course of 3 hours. So you see in this panel here, obviously we were hoping and expecting that there would be a reduction in the number of, in this case, orange and green dots over time. That would show that they were being removed. And then we analyzed each of these samples using something called a flow cytometer. It's a fancy instrument that just counts dots. So each of those dots is a single algal cell. And so these 2 types of algae separated really well in the flow cytometer. So each of those clouds is one type of these algae. And so the instrument takes those clouds and it tells you, you have 33,000 cells in this cloud. And then you can measure how that changes over time. So this is your first panel that I'm going to talk about. It was a really interesting one, really diverse. And this is, what this is going to show you is the reduction in the number of cells in each milliliter over 3 hours during the experiment. And so chlorella will be black shaded, and Synechococcales will be open, white. And you see both of these species were removed really fast. So within 40 minutes, it went from about 30,000 cells per milliliter to under 1,000. So really, really efficient, although organisms on this panel were just going to town on these algae. This panel was dominated mostly by barnacles, but if you could zoom in on that, you would see some little tunicates stuck in the crevices. So same idea. You see the Synechococcales decreased moderately over the course of the experiment. The chlorella didn't really do much. It stayed pretty much the same. Yeah?
So how does the bacteria increase?
So you're talking about like that 1 point in between 40 and 60? Well, so what probably happened is that the sample at it would have been the 50-minute time point was just, there might have just been something kind of off with that sample. And that if it was, that's one reason we took so many samples, is to kind of get just a general trend as opposed to just, looking at particular time points. It's a good question though. Yeah, there was definitely no increase in the numbers.
So it was a sample design.
Yeah, so this was a really massive experiment, and so we actually had 4 people pipetting at the same time for each of these, and so it could just be some kind of, a couple of the people were relatively new pipetters, so, yep.
Compared to these levels, what were the outer levels and the super level?
That's a super bloom. You multiply this by a lot, so it would be somewhere around, it'd be like 10 to the 6 instead of 10 to the 3, so it'd be a lot more, 3 orders of magnitude.
So I think the last one, it was the open box that was last one, is that right? In the last, sorry, the last
The
panel.
last slide?
Yeah, because it didn't die out completely, it kept existing. Yes, so doesn't that tell you, like, does that indicate that there's some species in there that's preferring to or better at predicting upon the shaded interval of species?
Yeah. So that's actually a really good question. Then I'll sort of get to part of that the last couple of slides, but then if you want to talk to me afterwards, I can talk to you about, or if anyone's interested, this is only part of this experiment, and so the second part of it actually allowed us to look at which members that were on these panels were removing chlorella or Senecio better than others. That's a great question.
There aren't the barnacles primarily zooplankton feeders anyway, so you wouldn't expect the alcohol concentration to go down.
Yeah, so you're talking about this one here.
Right.
Yeah, so barnacles do primarily feed on larger particles that are bigger than these, like zooplankton. And so the second part of the study and the analysis that I'll talk about in a minute do in fact show that the barnacles, higher barnacle abundance is actually correlated with lower removal efficiency of these panels. So this one was some barnacles and also some bryozoans. You can see not much happened. Now what we wanted to look at finally was how species richness, so once again that's the number of species per panel, how that correlated with the percentage of, this was the Senecio removed after 3 hours. We were hoping for a nice relationship, and we did see, this is a linear regression. We're currently reanalyzing these data, so but we're pretty happy with this, how this looks. So about 40% of the variation is accounted for by species richness, but I can't point at it, but I do want to highlight the samples. There's a group of samples up there, 1, 2, 3, 4, 5, at about 15 species per panel. Just remember those species for me. Remember those panels for me. So this is the same figure except for chlorella. You see another pretty good relationship, about 57%. But there's still these samples up here. And so what's interesting about this is, as your questions are really good about this as well, and it leads to a good introduction. But so you have these species on the upper right side that have, these panels rather on the upper right side that have really high species richness, between 30 and 20, and they have really good removal efficiency. But then you have these panels in the middle that have half the number of species and also have really high removal. And so it was likely that these panels had something on it that was doing a lot of work, being really, really efficient at it. So we ran an analysis, and I'm not going to talk too much about this, but it's a statistical analysis and also a way to visualize these data. So each of these dots is a panel. Each color is a site. I haven't talked anything about the sites, so you don't need to know about those. But this sort of topographical map, as you look across this, well, first let me say the placement of each of these dots is driven by the community composition, that's on each panel. And then this sort of topographical map here, it goes from, in your upper left side, that's a 20, and then on the right side is a 90. And so like that pink dot on the far left by the 20, that means that panel had 20% of its chlorella removed after 3 hours, so not very efficient. And then the dots over here on the right side with the red and the green, those had over 90% of the chlorella removed after 3 hours, so they're really efficient. And so what it says is that, so if we graph up how this relates to the abundance of specific species on each of these panels, we see this interesting trend where, so the arrows indicate increasing abundance of these 4 species on the right side. So the panels that had species that were really efficient at removing chlorella, generally had higher abundances of these 4 species, and all 4 of these species are tunicates. In some cases, the abundance of these species is really not that high on these panels. It was somewhere around like 20% on some of the barnacle ones in particular. And then A. eburneus is barnacles, and so this is the opposite. It means that panels that were not doing a good job at removing these were full of barnacles, just as we've discussed. And we do the same experiment with Synecocalcales. Once again, dots are panels. The placement of them within this graph is driven by what's on them, and then you have this topographical map from about 30% to 100% on the right side. And then you graph these, some of the species that are really driving these trends. Once again, all the species on the right side that are having a positive impact are all tunicates. And so these tunicates, at least in the Indian River Lagoon, appear to be doing, they're super efficient at, based on this correlation, it appears as though they're quite efficient at removing these particles. And once again, we did do a cool experiment. It's the second part of this that allows us to more directly look at which members of each panel were doing the removal. So I'd be happy to talk about that at the end. But yeah, so that is pretty much it. I would like to thank these people, and I hope that I sort of portrayed suspension feeders as interesting and also complex and sort of strange. So there's lots of funding that went into this, and I'd be happy to talk to anyone about that. But I also thank you, and I would be happy to take any questions at all. Thanks.
One thing that occurs to me is that if you had a biologically polluted waterway, you'd want to plant tunicates and not barnacles because they do a better job at cleaning stuff up. Is that generally true, or they only do a better job of cleaning some things up?
So I think that you could try to do tunicates. One thing that we've talked about with the Smithsonian is just because a lot of people in the Indian River Lagoon are interested in how it can be cleaned up and what can be done. It's a big, I imagine just like Kauai here, there's a lot of citizen science that goes on, and so people are really involved and they want to help. And so one thing that we thought about is just putting out more substrate where some of these suspension feeders can attach and clean just like they did with those panels. And so what they do currently is they take oyster shells and they put them in these small plastic containers that have big holes in them. People hang them off their docks with the hope that oyster spout will attach to the oysters and then eventually grow, and then those oysters will do their job. And it tends to, it's sort of mixed results. And so we wondered if there was some way to kind of provide more substrate in some of these areas that could allow these organisms to attach and do their thing. The problem's always money. So.
Yeah.
Very interesting. I had a lot of questions. I'm just trying to figure out which one to ask. But I don't know how much information you have or what your knowledge is of Hawaiian reefs and sponges in Hawaiian reefs compared to, say, Caribbean. But given our isolation and that these are, I guess, predominantly really shallow water organisms, how would they get to Hawaii, and what would be the comparative diversity and so on between, say, because you just look at the other aspects of this reef complex ecosystem, just coral or algal structures.
Yeah. I probably should have looked what the sponge abundance on Hawaiian reefs was before I came, but I think, and so we've been coming here a lot, and we've dove on all different islands. I don't think I've ever seen a sponge anywhere on reefs out here. That doesn't mean, so I've just been looking for emerging sponges that are obvious on the reefs. So I've been looking, a lot of sponges grow cryptically in little crevices and stuff on the reef structure. I do know that there's one area where there are sponges are in I don't think in the Pearl Harbor area, but in similar ecosystems on Oahu. Does anyone know if there's mangroves?
We have some mangroves.
That would be, if they're probably anywhere, it would be
mostly freshwater, red mangroves.
okay.
Yeah.
Yeah, I'm not really sure how.
A coral, for example, corals are very low species diversity here. And really no really strong reef-building corals in the water. There's only 4 or 5 really common species
Right.
because of the isolation.
Sure.
And I'm curious how sponges would go along with that. And what really is the job of a sponge on a reef compared with, say, the job of coral to build the structure and things like that?
Right, yeah, so I would say that the corals are more foundational species that are, like you said, being structured. The sponges on reefs here, I think that they'd be, I do know that generally they don't get the same size as they are in the Caribbean here.
Well, the water in Hawaii is very low nutrient as well compared to the top level and so on here. So that would be a big difference.
Yeah, I'll have to, it's a great question. I'm gonna have to look at what the verse is like here. But I would definitely imagine that's much lower than the Caribbean. Like you said, with corals.
A question about the sponges. They have these bacterial symbionts. How can you artificially move those symbionts or transfer them into a different sponge? So if you found a symbiont that was particularly good at fixing nitrogen, could you just introduce it into a different species?
You could try. People have tried, but I guess part of it is you just have to remember that these are like really long. The history of these symbioses go back many thousands, millions of years, and so if you introduce a species that might just be seen as either like food bacteria, so it might just be consumed without really establishing a symbiosis. But I think that more people, there's definitely research where they want to be able to do that, to kind of look at, so if I transplant these symbionts into this sponge, is it going to increase the host benefit, or is it going to improve the ability of that species to survive in some system? It's a great question.
Are sponges an old animal?
Yeah, so they're like you can think of them as like the most basic, sort of the first animal that came around. And so 600 million years ago, is when they first came about.
And do you have a sense that they did the same thing back then?
Yeah, so they've always been sort of this strange accumulation of cells that somehow all managed to work together. And so they have cells that the job of these cells is to bring water into their body and filter it out. And so that's what as far as I would imagine, that's what they've been doing the whole time. They've just been filtering water. I actually thought about showing a photo of them, but there are some carnivorous sponges in the deep sea that have these really crazy spicules, which are part of their skeleton. So like hooks, and so they eat like one shrimp a year, and that's all they need. So it's like they just wait for the shrimp to get caught, and they eat it, and then they're cool for the whole year. Yeah.
So when these sponges are filter feeding, what keeps them from consuming a bacteria that could become a symbiont versus a purely food bacteria? What's the mechanism there? Or do we know?
Yeah, so there's been some work looking at that, and so they think that it has to do with different metabolites or particles you can think of that are on the surface of the bacteria. And so some of them have receptors in the sponge that so it recognizes it, and it's like this is something I want to keep. This can be a symbiont versus this is something that I just want to eat, or something that I just want to pass through my body. Which, if you think about how simple sponges are, is a really complex idea, but there's definitely a lot of aspects of sponges that are much more complex than you would think, just based on how simple their bodies are. But they don't have any brains or anything like that, any eyes, or so.
That's pretty creepy. You come to visit the sponge, you don't know whether you're dinner or staying by dinner.
Yeah, that's true.
It produces spicules, and that's their basic rock record. Geologic record. A lot of people wonder what the hell they were when they first started identifying these things.
Yeah, I mean, you can. Have you ever, anything about using sponge spicules to look at geological history?
A little bit, yeah. There's some microbial things like that, microfossils that are used to correlate. And there are some variations from that.
Okay.
So, yeah, that's part of the rock record. They're not a huge volume of hard parts like coral or coral algae or something like that, but you find them in sediments.
But how, well, we can talk after you on, but would you be able to identify like, general groups of sponges if you had someone who really knew their spicules?
And they also there are some sponges that are biodegraders in terms of the reef itself, and more climate effects.
Sure.
That's one of the main elements that destroys the reef. So there's a balance in there. So.
Yeah, there's a lot of Caribbean corals. If you know, a lot of them are super degraded anyway, and so if you break off a piece of it, you break it open, it can just be sort of tunnels, and inside the tunnels might be like a red, very thin red flesh, and that's all these borean sponges that really tear up a reef.
What's the mechanism that the sponges use to move water? I mean,
So they have
pumping somehow.
Yeah, so they have these little cells called choanocytes, and it's just like it's a cell that has a ring of cilia that they use to filter, and it has a flagella, and so it just moves back and forth like that. And so it's an incredible amount of coordination for an organism like that, but somehow a sponge that has thousands, tens of thousands of these cells can make them all go at once to bring water in. If a sponge gets clogged up, we can actually reverse flow to try to spit the sediment or whatever's coming into its canals back out. So that's what's on.
It says that without a brain.
Yeah, without a brain, exactly.
On the topic of tunicates, they have an alternation, they have some sort of life cycle where they're not sedentary for their whole life cycle, is that correct?
Yeah, so both sponges and tunicates have sort of a larval cycle where part of them is going through the water column. So it's one way that when I showed you those panels, how those organisms have settled on those. So these are the organisms that originally settled on those panels were sort of this mobile stage of the life cycle of those organisms. Yeah.
They tend to be sessile.
Yeah.
So as far as them actually getting into Hawaii, they would have to attach to something that floats.
Right. They can be invasive, in like ship ballast or travel that way. Or they can,
before humans arrived here, the native sponges.
They can attach to something and make it potentially, but
it would be a rare.
Yeah, I don't think that there would be any. It would be really tough for a larvae to survive a trip from, I don't know where it would come from even.
That's why we don't have very many corals here.
Yeah.
Same problem.
What's the reproductive rate?
So it varies for each species. There's actually not much known about sponge reproduction, but for the most part, it's thought that one idea is that sperm is released by one sponge, travels to the water column, is taken up by another sponge, and then fertilization happens inside that sponge. Sort of a weird process that happens there, and then the larvae is ultimately released from that sponge, and then that larvae can travel in the water column to reach a different site, or it can settle out nearby, or it could travel for a few days or maybe longer. But sponges are really hard to work with in general. They're really finicky. They're not like corals. Like a couple of my buddies study corals. They'll go out and they'll want to collect corals to bring back to the lab, and they'll get corals and they'll wrap them in like bubble wrap with no water and put them in a cooler, and then they'll take them across Florida, put them in an aquarium. When they get there, the corals are up and they're perfectly happy. And like sponges, if you are in the Caribbean and you collect a sponge and you take it out of the water for like a second, like just to move it from one bucket to the other, it's dead, and that's it. So it's really tough to do experiments with.
How are sponges reacting, if at all, to the stresses
In general, it's thought that they're more robust than corals because, especially in the Caribbean, their abundance is actually increasing in the Caribbean. So there's a relationship that's, corals are declining and sponges are increasing in abundance. And part of that could just be due to the way that they eat, and also could potentially be due to their interaction with their symbiotes. But there's a lot of work looking at like ocean acidification and increased nutrients and increased water temperatures. But it sort of varies across species. I think some sponges are impacted more than those things than others. So it's tough to sort of get a general summary of it, but it's I think overall they're doing better than corals, so that's good for them, but still not good for corals. Yeah.
So is there any evidence of them picking up microplastics or small?
Yeah, that's a good question too. So a lot of people in the States are studying microplastics in different organisms, and so there has been some work on if you can you can actually dissolve away the sponge tissue and it leaves the microplastics that are present in it. And so I've seen some photos from a poster where they took a decent sized sponge, maybe this big, dissolved its tissue away, and then they were left with like a petri dish of microplastics. And so they do become embedded inside the tissue. And whether or not that hurts the sponge, who knows? But it could certainly clog the canals or something like that, make it difficult to filter and function.
How did you get interested in sponges?
It's a weird organism. So I was an undergrad up at Connecticut College, and I knew I wanted to go to grad school, and I just read this paper about sponge predation by parrotfish and stuff like that, and I was like, sounds really cool. And so I applied to some different schools where I could study that, and so I worked in a lab up in Georgia, in the United States, for about 2 years, studying that off the coast of Georgia. And then so I just got, after that I got really into sponges, and then I ended up doing all my work on them.
Parrotfish eat sponges?
Yeah, there's some parrotfish they eat some sponges. They eat sponges that are sort of not defended.
Do people eat sponges?
Yeah, there are some people in, I think it's Greece, there's a very specific species. If I have it right, it's like the grossest species you can think of. It's like really rubbery and sort of slimy. They put it on salads, apparently. I would suggest it.
How do they taste?
I would not. Some people say that's a science you should eat your organism, but I think it's not.
Are there toxic sponges?
Yeah, so sponges, there's a lot of work that's been done with chemical defenses in sponges, and how some of them are really defended with these really potent anti-feeding compounds that deter fish. So if the fish bites it, it just tastes gross, and so the fish swims away. But sponges are actually one of the most likely sources of like biopharmaceutical products. So there's been some work that, I believe it was AZT, which is the first HIV drug, came from a sponge. And there's some sponges that are being tested at Harbor Ranch, which is in Fort Pierce, where I did my postdoc, and they're in clinical trials for pancreatic cancer use as a drug right now. So, because nature really produces compounds that are way more complex than humans can come up with themselves, and so they have these unique properties and activities that we wouldn't think of. And so, there's been a lot of work looking at sponges, tunicates, pretty much anything from the sea has been collected, and as much as possible, they collected and analyzed it to see what they can do.
Thank you.
Thank you.